Hi everyone. Thank you so much for inviting me here today to present my research. As I said, this is my first research grant, so I'm really excited to be working to try and develop some chemical probes for Alzheimer's disease. So my work is focused on developing inhibitors or degraders for tau K1 through 3, which I'm sorry sounds really similar to tau, which is implicated in Alzheimer's disease. So I'm going to talk to you a bit about that today. Next slide, please. Okay, so tau K, which are kinases implicated in Alzheimer's disease, are overexpressed in AD patients' brains. They are activated in Alzheimer's disease and they co-localize with phospho-tau. They're also activated by tangle-bearing neurons, so there's kind of a feedback loop of activation when tau K is involved. And they actually phosphorylate microtubule-associated tau, which leads to destabilization, tau aggregation, and then the formation of neurofibrillary tangles, which I will only say once today, and now I'm going to call them tangles. So they're heavily implicated in Alzheimer's disease, which is why they're a really interesting therapeutic target. So on the left just highlights this pathway some more. So phosphorylated tau Ks activate tau by phosphorylation, which leads to the microtubule destabilization, and then tau aggregation. So when that occurs, that's one of the key hallmarks of Alzheimer's disease. So with this research, we're trying to stop that from happening. We can do that in a variety of ways. We develop inhibitors, or what I'll call PROTACs throughout this talk. So they are proteolase targeting chimeras, and they actually are degraders. So they can degrade the protein rather than just inhibit the function of it. So on the top, you can see I'm inhibiting tau K, and on the bottom, the protein's completely gone. So we're hoping that by developing these methods, we can reduce phospho-tau, cause microtubule stabilization, and even reduce tau aggregation, which could be beneficial for Alzheimer's disease patients. Sorry. And I mentioned in the first slide that I'm developing a chemical probe. They're slightly different to drugs. So drugs must be safe and effective, and they're useful for clinical validation within disease. Whereas what I'm developing is very early stage, so it's used to ask a specific biological question within disease. So they're slightly different, but they're also extremely useful. And what we do actually in the SGC at UNC is we're part of open science. So everything we do gets published. There's no IP. So once we publish these results, anyone can use that data to improve the science further. And with this project, we'll be looking at tau K1 through 3 inhibitors and degraders and seeing if they can help to reduce tau aggregation. So apologize, this slide might be a bit difficult to see, but CP43 is the current inhibitor that's in the literature that's used to target tau K1 and 2. And it's very potent against inhibiting the function of tau K1 and 2. So on the top left in the yellow box, on the top, you can see there's a little red dot, and when they add CP43 into these primary neurons, that little red dot has disappeared, and that's phospho-tau. So using an inhibitor can decrease phosphorylated tau, and then at the bottom in the yellow box here, you can see that the pink blob has disappeared, that's a result of actually removing tau K2 entirely, using a method called shRNA, which is an artificial way to remove the protein from the neurons. So this kind of validates the improving inhibitors that are out there and actually developing the first PROTAC to try and degrade tau Ks 1 through 3 could be beneficial. And this indicates that tau K2 could be the protein we're interested in, but tau K1 is also implicated in these pathways as well. So really having tools that we can better understand are really useful. So this is just an overview of what tau K inhibitors do and what they look like. So on the bottom left, tau K proteins use ATP in the cell to convert that to ADP. Basically what this does is it kind of flips on a light switch in the cell. So substrates are activated by phosphorylation with the presence of activated tau K, and that leads to downstream cellular responses such as phosphorylation of tau, and there's a myriad of other responses that occur when a kinase is activated in a cell. So traditionally with inhibitors, we focus on this site where ATP binds to try and inhibit it, which makes it incredibly challenging because there are a lot of kinases and ATP binds to all of them, so they're very structurally similar. And on the right, I've shown tau K1, 2, and 3. And as you can see, there's not a huge amount of difference between all three of them. Yet we can still actually design inhibitors that would target tau K1 over tau K2 or 3, for example. It's just a little bit challenging. So this is just an example of why degradation is good in relation to inhibition. Inhibition's great too, don't get me wrong. So on the left, when you inhibit a specific pocket, such as the ATP binding site, you actually just remove one biological function of the protein, whereas if you degrade the entire protein, essentially you can remove all the multiple other functions that the protein may have implicated in disease. Additionally, PROTACs are kind of useful because they can actually degrade any proteins that are mutated, which is very common within disease, and they can impart greater selectivity of degradation over inhibitors. Next slide, please. So this is how PROTACs work, and I'm not expecting you guys to look at every single aspect of this, but I'm giving you a broad overview. So that little, the two dots on the right attached with the squiggly line is what I use to show what a PROTAC is. So obviously it's a lot more complex when we look at the chemical structure, but this is just basically designed to tether two proteins together. So what I do in the lab is I synthesize those PROTACs, and then I dose them in cells. Can we move to the next slide, please? Sorry, this is an animation. And then that brings the E3 ligase, which is part of a machinery that leads to, it's involved in degradation, and it brings that into proximity with the protein we want to degrade. So you can see that those two proteins are brought together. It transfers something called ubiquitin, and then that tags it for proteasomal degradation. Next slide, please. Yeah, so then the PROTAC's degraded. It's pretty complex, unfortunately. And there are lots of things that can go wrong along the way, which is why designing them and testing them is pretty challenging. Move to the next slide. But the benefit of PROTACs is actually once it's degraded some of the protein, it's released and it can go on to do it again, which is actually really beneficial in terms of dosing. You can actually put less on a cell than you normally would because it can catalytically turn over. So now on to my project goals that I am working on currently. So the first goal was to develop a cellular assay to determine the potency and selectivity of these chemical probes in cells because they currently don't exist. And it's really important when you're characterizing all of these compounds to see what else they do in cells if they engage with your target. Next slide, please. And then obviously to degrade PROTACs, you make PROTACs to degrade tau K1 through 3. And this is an example of the chemical structure, not one for tau K, but just so you can get an idea of what they look like. And then the ultimate goal is once we've developed these PROTACs that degrade tau K1 through 3, we want to test them in neurons to see if they can actually have an effect on decreasing phospho-tau, and if it's more effective than just inhibition alone, which we're hoping it is. So this is just an overview on why we're interested in selective probes. So when we think of a tau K inhibitor, we've got three family members, tau K1 through 3. There are actually more proteins that are involved in that family. But when we just look at 1 through 3, for example, if you inhibit the kinase domain of all of those proteins, you don't really gain any specific information about any singular biological function. Whereas if you had a specific inhibitor that just binds to tau K1 and inhibits that activity, you can now actually use that to understand the specific role of tau K1 in disease or tau K2. So having a selective tool to study disease is really important for understanding the disease better. And then with kinases, unfortunately, it's not just 3 kinases that we're looking at for selectivity. There are actually 538 kinases, human kinases. So we have to then look at the selectivity across all of them before we can really say we've dosed this compound in the cell, this effect is a result of inhibiting this protein. So we actually have a number of assays to do this that are biochemical and in cells, and they're really essential to trying to assess the selectivity of these compounds. So this is a bit of a scary diagram, but it basically represents all of those 538 kinases that we look at when we try to determine the selectivity of these compounds. You hit the next slide, please. And we actually have a cellular assay developed by Promega, which is called a K192. And all of these little red dots here are the assays that are enabled within this screening set. So basically, I sit there and I run 8 96-well plates. It takes a long time, and each well has one of these kinases in it. So I can test a compound against all of these kinases to determine the selectivity in cells. So that assay is called a nanobret, and it's a little bit complex, but we transfect in our tau kinase with a nanoluciferase, that little blue glowing thing there. And we also add in a tracer, which has an inhibitor for tau K and a Bodhi. And when they're in proximity, we get a signal in BRET, is what it's called. And then if you add an inhibitor, you can feed it off and actually determine the cellular potency of compounds. So we do a lot of work making these tracers and trying to enable new nanobret assays in the lab. And until now, there was no assay like this for tau K1 through 3. So this is data for my tracer FP145-1 that actually enables an assay for tau K133. So the most important thing to notice is that the pink line going up is when we add in the tracer. So we get an increase in this luminescent signal, and we can remove it to have no BRET when we add in the inhibitors, which is the black line. So there's a window there that we can use to actually assess our compounds. A small caveat, this assay is actually not in fully intact cells. We have to use digitonin to kind of, it pulls open the pores slightly so that tracers and compounds can get into the cells, but it's still a great step in the right direction towards assessing these compounds in cells. So these are the results from assessing the top two inhibitors, and then the next ones are proteins that I've synthesized in cells in that assay. So green is good. It means that it inhibits the assay, the kinase pretty potently. So smaller numbers are better because it means you use less of the compound to inhibit the function. And then red is bad. So it's kind of a traffic light system. And within the lab, before I even joined, we actually had a postdoc called Alfredo who had made a compound AP32, and it is pretty good at inhibiting tau K1, 2, and 3. And it appears to be slightly better than CP43 that's in the literature right now. So you can see on the top there's a bit more green going on there. And then I assessed it in this kinome-wide assay. So what you're seeing on the right now, the red dots just indicate kinases that are inhibited. So you want to see as little red dots as possible for it to be good. Tau K1 through 3 is obviously not in this panel because no assay existed. So what you can see here is there are a few red dots, which is really good. And then when I followed them up in the nanobret IC50 shown in the right-hand column, you can see that they're not very potent. You want a number less than 1,000 to be cell active. So basically, this inhibitor is pretty selective towards tau K1, 2, and 3, which is a really great step in the right direction. I also screened CP43 from the literature, and as you can see, there's even less red dots, which is pretty promising, although it is slightly less potent against tau K1, 2, and 3. So there's further work needed there, but we've got some interesting lead compounds to try and develop into chemical probes. So now I'm going to give you a very brief overview on how I synthesize PROTACs. So we start with an inhibitor like CP43 and AP32 that binds to tau K1 through 3. We then need an inhibitor for an E3 ligase, shown in number 2. This is extremely well precedented in the literature, and there are loads of ligands that bind to multiple E3 ligases. So we picked 2 that were commonly used. And then in 3, you can see I've attached a little squiggly line in between. That's me just going in the lab and adding a little linker region into them to bring them, to link them together. And then we test them in cells and try and assess degradation. So over the last 10 months, I've synthesized about 14 PROTACs in total. And if you move to the next slide, I'll show you how we then assess degradation. So quite commonly you do Western blots, which is you would dose them in cells and then run gel and an you'd use an antibody to look at them, and it's really time consuming. I spend a lot of time in the lab doing Western blots. So this is a high throughput method that was developed by Krumega. You start off with your cells on the left, and you transfect in this tau K133 with high bit, with that little blue piece of pie on it. That's the high bit tag. And then you also add in your degrader. And what you see, if it does degrade that protein, is you get a loss in the protein that's tagged with that little piece of blue pie. And then after your desired time point, you would basically lyse the cells, which is just busting them open, and adding in this Pac-Man kind of LG bit, and that attaches to the pie, and when that's together, it generates a luminescent signal. So if you have lots of that left over, you get a strong luminescent signal, and if you don't, it decreases. If you could hit the next one, please. So this is just an example of what you expect to see in these kind of graphs. So a DMSO treated with 0% degradation is a control that you should never see degradation on. And as you can see, that's at 100%. And if you've treated it with a degrader that works, you also get a decrease in the signal because that protein has been completely removed. Move to the next slide, please. And this is horrific, I'm sorry, there was no easier way to show this. But the top line is tau K1, the next is 2, and the bottom is tau K3. And when you see degradation, it should fall below that blue line. And intriguingly, with 3 of my PROTACs and actually 1 of the inhibitors, we do see a substantial decrease in the protein. And I've highlighted them in green on the left there. So you can see that quite a lot of the concentrations go below that blue line, which suggests we're getting degradation of, in some cases, all 3 of these proteins, and in others, just of tau K1 and 2. So the next slide has these 4 kind of highlighted a little bit differently. So 1, 2, and 3 is indicated in blue, red, and the yellow. And this is over 2 biological replicates, so we know that it's robust and it repeats. And then the green lines that I've added on the right are for viability. So you can often get a decrease in this signal just because the cells are dying and they're unhappy. So we do a control where we just test how happy the cells are, basically, for viability. And if you track those green lines across, we want the bars in blue, red, and yellow to be below that green line. And as you can see, in a number of these inhibitors and PROTACs, we are getting substantial degradation that's not a result of cell death. So it's likely that these PROTACs are real. So in summary, I developed an nanogrit assay to study tau K1 through 3 in cells. I've also assessed the selectivity of AP32 and CP43 in a wider cellular context. I synthesized 14 PROTACs to try and degrade tau K1 through 3, and luckily I found at least 3 so far, which is good. And then also serendipitously AP32 looks like it's acting as a degrader too, which is really exciting. And then obviously there's still a lot of future work to do related to this. We still want to improve the nanogrit assay to see if it works in intact cells. We need to do a lot of control experiments with the degraders just to show that they are in fact doing what we think they're doing and how they're doing it. And then we also look at the entire proteome. So we'll take a cell and we'll do mass spec and see if it degrades anything else. And we're also then going to move into looking at what happens to phospho-tau and tau aggregates in neurons. So that's the end goal now that we have those PROTACs. So very appreciative of Dimensional Alliance and Guardian Angel Thrift for providing the funding to do this research. It's really important. I can't do it without funding, so very, very happy that you guys chose to fund my research. I'd like to thank David Drury, who is my mentor for this project, and everyone in the lab that had worked on it. We're part of the SGC, which was shown here. We're quite a big lab. I'm one of 5 PIs, so there's a lot of support where I work. And yeah, if you've got any questions, I'm happy to answer them. Yeah.
So the towels, are they selected from what? From like mice or human? I mean, what are you testing these biologicals on?
Yeah, so tau K1 through 3 are present in all cells, mice, humans, but in my lab we just look at cells for now, and then, you know, essentially if someone was able to, they could go and look at them in mice, essentially. They're present in in mice, humans, and cells. So, yeah. Yeah, but my lab predominantly just works in cells. So, yeah.
So I know when we were looking at your project in particular, it was talking about how this would have, you know, really good application for future researchers to be able to use that. Can you talk a little bit about how someone, once you have completed your project, would be able to share, say you were published and share that information, what that next step is to the process, whether it's with you or another researcher?
Yeah, absolutely. So,
Can you
yes.
speak up a little
Oh,
bit?
yeah, sorry, sorry. Yeah, so once I've developed these PROTACs and inhibitors, my plan is to publish them, and then anyone that sees that can collaborate with me. I share the inhibitors and PROTACs with anyone for free. Essentially, I'm very happy to collaborate with anyone. So labs that are working on Alzheimer's disease that do have those mouse models and all those studies would be able to test the compounds in that, and having it just in the public knowledge. I mean, there's no IP on this at all, so when I publish it, anyone from industry or anyone that's working on it can take it and they can make it better. In some senses, that includes just kind of, we're working in cells. There's a lot of work you have to do to make it work in mice. For example, there's some modifications that the compounds need, and that's some stuff that we do occasionally, but there are lots of labs that are very well equipped to do that as well, and try and transition that towards more of a drug rather than a tool.
So you're kind of narrowing it down.
Yeah, yeah.
So I have a daughter that's getting ready to go to college, and research is certainly a part of what she's thinking about. So when I had the opportunity to go visit with you, 3 months ago now, I think it was, there was a whole bunch of people in that lab. How many people actually worked on this project? And were there any students, interns? How did all that work on this project?
So most of the work is actually mine. Yeah, I don't have enough funding for students yet, but I hope to. Obviously Alfredo, he designed the compound initially, and there's a large portion of data that I didn't show related to that. But even though there's lots of people in the lab, this particular research was all done by me without any help. So I'm hoping to get some help soon.
I will have to say Dr. Bashore called or emailed me yesterday and said, hold on, she got me the PowerPoint late yesterday because some of the results were coming in as late as yesterday afternoon. So this is fresh information that we were receiving today. So thank you for doing that.
Yeah, no worries. We just got the degraders yesterday, so wanted to share that with you guys.
Any other questions for Dr. Bashore? No? Well, I have a little thank you for joining us today, and thank you